Hi, good afternoon. It's a great pleasure to be here with you. Okay. From cars to cell cultures. That's a huge ride, actually. This is the biggest challenge I will have today. Um, okay, let's start from the very beginning. So, mentioning the cars, we won't go far with clinical trials or with personalized medicine without cell cultures. So this is our base, actually, to our new drug, new therapies, tissue engineering, and whatever else we would like to imagine having in the cells in mind. But except for these key sectors, of course, we have key drivers on the market that will push this cells monitoring forward. And for example, here, I think you heard about reducing animal testing, right? So if we will reduce animal testing, so the step in between for clinical trials, we have to go like one step back to this cell culture, so the first step. And we will have even even greater expectations, and we need much more information from this first step. That's why we need to have a new technologies, new system to be able to monitor them in real time. But before I will show you our system, because today I would like to introduce to our new technology that I'm developing with my team, one quick thought from the very good book actually that I read some time ago. It's the Song of the Cell, and one thought that stayed with me is that everything starts with cells, not with our body, not with the tissue, not with the organs, but if we would like to find the source of the sickness, of our health, longevity, and so on, we should start from the single cell. But okay, what's the for those of you who are not familiar with cell cultures, especially, what's the biggest challenge here, actually? So here we have 2D culture, very simple cell line, creating a layer on the cell dish. But what you can see, this is a very dynamic system, very complex. And these cells are moving constantly. They will reorganize, they will proliferate, they will bind with each other, yeah? So we can see that from the measurement point of view, from the engineering point of view, this is a huge challenge because we will have a constant change. We need to be able to track those changes in real time. But how it looks like right now? Imagine you would like to test a new drug or new therapy. Yeah? So we have a few cell culture dishes, probably many. And then you have, of course, specialists who run multiple tests, biochemical one, some visualizations, and he or she will take some samples from the cell culture dishes and run those tests, right? But here, the information we will obtain depends entirely on when we will take this measurement. So knowing now how dynamic this process is, do you think it's enough? Like these few points that we take during this cell culture, like we can miss so many different stages, so many different important points actually in the culture. Like we can miss even the point where the drug is starting to work, yeah? So I like this metaphor that doing that kind of measurements, that kind of tests, is like trying to understand a movie basing just on few snapshots. So I will leave you with the question, is it possible to understand that kind of process with this methodology? Of course, we have another methodologies, because cell cultures, like I said, these are very basic systems for clinical trials and other. We have also microscopy, very well established. But for microscopy, we need some dyes, we need labels, some markers, fluorescent one. But the problem with this is we have to add something to the cell culture. And sometimes we don't want to change our culture, our cells, right? We would like to monitor them as they are, because in our system, in our body, we don't have these labels. And for some cells, of course, they will be perfectly fine with these labels, but some of them may be changed a little bit, but still, yeah? And the problem, the next one, is that if we are talking about monitoring cell cultures, we are not talking about days and few tests. Sometimes we are talking about weeks. So after weeks, we don't have any fluorescent label anymore, and we are not able to track it. So going even further, for another technology, we have also some electrochemical systems. But again, electrochemical system, here you can see the plate. Okay, it's not working. We have a plate with gold electrodes, and here we can take some measurements, but if we would like to run electrochemical tests, we have to electrically stimulate some cells. And again, for some of them, they will be perfectly fine. It's non-invasive, but for some, they can be changed. So we would like to avoid it. So what kind of system we need? It should be continuous measurement, should be label-free, now we know. Non-invasive, of course, capable with the workflows, because we don't want to change entire lab. We would like to have additional system. So our question was, can we design a sensing system that follows the natural behavior of cells instead of interrupting it? And here's our sensing concept that I would like to introduce you to. It's microcavity inline Mach-Zehnder interferometer. I know the name is very complex, but I promise you don't have to remember that. This is the cross-section of the optical fibers. And optical fibers, you are familiar with them in terms of internet, data, and so on. But here, we will use it as a biosensor. So instead of the fiber, you can imagine your own hair, just one single hair, because the size is very similar. And then imagine that someone drilled a very small hole inside this hair. And this is our sensor. And everything what will happen inside this microcavity, we will see on our signal. Of course, if optical fiber, we will have the light passing through the fiber. Then it's an interferometer. So at the end of the fiber, we will have the signal. What I need to mention here, we don't have to understand this signal, but what is very important, it's extremely sensitive to what is happening inside. And here are some other photos. And what we did actually till this time, we proved that we are able to follow this very dynamic process of adhesion of cells. So as you can see here, it's not stable at all, yes? So to be sure that every single change of the signal is not instability of our sensing systems and so on, we of course added other different methods to it. So we are able to create the software, data analysis, and to provide actually ready-to-use product to your labs, hopefully. And what is great about fiber optics is that it's just glass. So it's highly biocompatible. We don't need additional layers. We don't need collagen, polylysine, other thin layers. As you can see, cells very likely grow on it inside the microcavity, on the fiber. There's not, we don't have any problems with that. But, of course, here I talked about many advantages, but of course we have some limitations as well. And one of the biggest limitations that we have right now is the size of the microcavity. Because, as I said, everything what is happening inside I can see on the signal. But if something is outside of the microcavity, we are not able to track it. So if the cell is bigger than the microcavity, we won't use it. So now we are testing different sizes, different fibers, different materials. So at the end, we are able to build our prototype. So you can imagine it as a 96 well plate covered with embedded optical fiber sensors. And I hope in 2 years I will be able to come back here and show it to you with the first version of the software and data analysis, which we are working on right now. And what we are planning to use it for is the applications that we are not able to use any other sensors. So now we are working on radiotherapy monitoring, for example, with gamma radiation. Other sensors cannot be used during this radiation, but our optical fiber is perfectly fine because it doesn't react with, during this radiation. So everything what's going on inside the microcavity, we know it's coming from cells not from irradiation. We would like to check also the cell therapy monitoring and real-time biocompatibility tests. So if you work with cells, if you are willing to try new methods, and if you feel that you need some more methods to monitor them and track or find some special point in your cell culture, just please let me know. Let's stay in touch here or on LinkedIn, on my email, and yeah, thank you so much. No. I see that we have 2 minutes for some questions. Do we have some? No?
Our plan, we always do it 25 minutes.
No. No. Okay. Should be up, like on the.
She said yes, that I can open it questions to them.
No. Okay. Hello.
Hello. Yeah. Thank you for the nice presentation and your topic. Actually, I am a PhD student, so I have a little familiarity with, but not exactly with biosensing. But I would like to know that how exactly you did the measurement. For example, in the 96 well plate, in terms of an experiment, we use ECIT in the instead of we use cytometers or spectrophotometry, or in that terms, could you please tell me if I can understand?
So for now, we are 3D print our cell culture dishes. So we embed the fiber inside the cell culture dish, or we are putting the fiber on the bottom of the cell culture dish and put the cells on the fiber. So the cells are always inside the microcavity. They have no problems to go inside, if of course the size of the cells are right. And yeah, and this is how the measurement looks like actually. And then we monitor how the cells behave based on the morphology, movements, and yeah. So it's pretty simple actually. Okay, thank you so much. One more time.